All right, welcome back. So in this part, we are going to discuss what are known as the tunics of the eye. And when we talk about this term, tunic, the tunic is essentially a layer. Okay, so we're gonna, in other words, you could call this like the layers of the eye. All right, and so we'll see three general tunics. All right, and then we'll kind of name these tunics. We'll name them something specifically, and then associated with each tunic or with each layer, there are certain structures and there are certain features. So these three walls, these walls are formed by these three tunics, these three layers, and they are known as the fibrous or the external or the most superficial layer. We have a middle layer known as the vascular tunic, meaning it contains lots of blood vessels. And then we have an inner tunic or an inner layer known as our retina. Okay, and so for each of these, there will be subcomponents or substructures that we will discuss related to each one. So for the fibrous, we'll talk about something known as the sclera and the cornea. For our vascular layer, we'll talk about something known as the choroid plexus, or not the choroid plexus, I'm sorry. We'll talk about the choroid. We'll talk about the ciliary body. We'll talk about something known as the iris. And then for our retina, we have a what's called a neural layer, and we have something known as a pigmented layer. And we'll talk about all of these in a little bit more detail. But in this image of the eye here, you can see these layers. So this outermost one here, if you follow this, this is our sclera and our cornea, making up our fibrous tunic. In the middle, we have this darker kind of red-pink vascular layer associated with this iris, choroid, ciliary body. And then we have an inner neural layer, which is primarily what we think of as our retina, right? So you've heard of your retina. You may have heard of a detached retina. That's this inner layer, this inner tunic of the eye, okay? So when we look first at our outer layer, we look at this most superficial layer, our fibrous tunic, okay? It is composed of the sclera and the cornea. Now, the sclera is very easily seen on your own eye. So on living specimens, you can see this very, very easily. It's simply the white of the eye, right? So when you look at somebody's eye, or if you look at your eye in the mirror or a picture, the part that is white, that is our sclera, okay? It is composed of connective tissue, so primarily dense irregular connective tissue. So if you recall, when you're talking about histology, dense irregular, those fibers kind of running in kind of a random orientation. Some are going vertically, some are going horizontally, some are going obliquely, right? So they kind of go in this in these multiple directions. This helps contribute to our eye's shape, and so this form, this kind of bulbous, kind of round structure, and it's also relatively firm as well. And so this helps to protect from trauma or from any kind of external things in our environment that would penetrate the eye. It serves to protect it and protect the inside of that eye. Now, so that is the sclera. So the sclera is all of this kind of more external white of the eye. As you run onto the anterior surface of the eye, so you start out here laterally or superior or medially or inferiorly, and you run into the middle of the eye, right? So where we have our iris and our pupil. On the anterior surface, you have something known as the cornea, okay? So the cornea is that anterior kind of covering, this kind of curved convex covering over the anterior surface of the eye. So if you look at the structure of the cornea, let me go back. If you see the structure right here, this is our structure of the cornea. So you have these various layers that are on the superficial surface. Now, one important thing to note about the cornea is that it does not contain any blood vessels. Okay, so it is avascular. It receives its nutrients and oxygen and whatnot through diffusion from the tears. So as the tears come across the surface of the eye, the nutrients and oxygen and gases and whatnot will diffuse into the cells making up the cornea. But the cornea itself does not contain any blood vessels. So think about it. So think why would that be a beneficial thing? Why would we really not want blood vessels in the cornea? Okay, and so another thing about the cornea to keep in mind, this is where light is going to first pass through, right? So if we represent light by this orange drawing here, this is a wave of light, right? As it's coming down, it is going to pass through the cornea first, through the pupil, before it gets to our eye. So you may think, well, if we had blood vessels running in the cornea, that blood vessel in the blood may deflect the light, and it may obscure it, and it may prevent the light from reaching a certain part of our retina. So the lack of blood vessels actually, it is a good thing because it is preventing light from scattering preventing light from hitting the blood vessel, hitting the blood, and refracting kind of all over the place, and that would make our image that we're seeing, it would make it very unclear and very fuzzy. Okay, so this is what we're talking about here. So we're getting our nutrients from the tears and the aqueous humor. We'll talk about what this is, and we lack those blood vessels here. Okay, so this also has an implication for corneal transplants. Okay, so you may have heard of a corneal transplant, or somebody donating their corneas after death. The cornea is the most commonly transplanted structure in the body. And one very good thing, and it'll make sense now that you know that it's avascular, one very good thing about it is that you don't really have to match up donors in order to receive a cornea. In other words, with lots of other structures, you have to make sure the donors are compatible in terms of their surface proteins to prevent immune reactions. Well, if our cornea does not have any blood cells, and so the basis of an immune reaction or a transplant reaction is the basis of that is white blood cells coming from the blood to that new structure and attacking it, right? So the host white blood cells attacking that new structure. Well, if we don't have blood vessels, then there's no way we can bring white blood cells there, so there's no way you can have an immune reaction. So you don't have to worry about a rejection, the host rejecting this graft or rejecting this donated cornea, because there's no blood vessels that could bring white blood cells to attack it and to destroy it. All right. So we also see the cornea here. As it is passing through, it can refract the light. In other words, it can essentially bend the light and focus it on a specific region. Now, we also have to work in conjunction with our lens, and we'll talk about that. But you can see here, as light is coming in, it's passing through the cornea. It's slightly refracting and bending, hitting the lens, and then that lens bends even further. And then we come back here and we focus on a region of the retina that we'll see in a little bit, known as the fovea centralis, which is essentially where we have our best central vision and our best color vision. Okay, but keep in mind, light has to pass through the cornea, and as long as light is passing through this cornea, and then it can be refracted onto the retina. Okay. All right. So here you can see our general location of some parts of our eye, and you can see some of the parts of this fibrous outer tunic. Right. So you can see blood vessels running through the eye. This is a more pathological condition, or this is something that is more, in other words, it's not a normal state here on the right where you can see all these blood vessels. But if you follow it onto the anterior surface of the eye, you do not see any blood vessels in this region here, and it's because there's not any, right? We said we do not have any blood vessels associated with our cornea. All right, so if we move past the outer fibrous layer, right? So we talked about the fibrous layer first. We move just internal, just deep to that, and we get to something known as our vascular layer, or our vascular tunic. All right. The term vascular is referring to the fact that we have lots of blood vessels here. Okay. So this is composed of three different regions or different structures. One is known as the choroid, one is known as the ciliary body, and something known as the iris. Right. So of these three, you've probably heard of the iris. Okay, so the iris is that colored part of the eye, and we'll talk about that in a second. So the choroid is this middle, kind of this stretching middle layer of the eye. You can see right here. So you have your outer fibrous layer here, you have your inner layer right here, and right in the middle, right where I'm kind of drawing right here, this is our choroid. All right, so it contains lots and lots of blood vessels that nourish our retina, so it has a large capillary supply. And it also contains pigment-producing cells, so we know these as melanocytes, that can absorb extra light and prevent it from scattering throughout the eye and thus causing us to see a distorted image of whatever it is we're looking at. Okay, so you can see the same thing down here. You have an outer fibrous layer, right? So you see the sclera here. When you look on the inside, you'd have the retina deep within, and then you'd have the choroid right here. Okay. All right. If we continue looking at our vascular layer, we also see something known as the ciliary body. Okay, so the ciliary body is this very large kind of circular structure that contains something known as the ciliary muscles and ciliary processes. Okay, and so you can see the ciliary processes right here. We can see a large ciliary muscle kind of all in this region here. Okay, so both of these kind of combined together are known as the ciliary body. All right, so what the ciliary body and the ciliary muscles do is these help to adjust or to change the shape of what's known as our lens. Okay, so the lens is what is really focusing and adjusting the light and focusing it onto the retina. All right, so hopefully you can see here, if we're looking at this picture up here, we see these muscles here. So we see these muscles that I've circled here. You see these small little processes here, and then you see these fibers stretching out. All right, so as the ciliary muscles contract and relax, so they're a smooth muscle. All right, as these contract and relax, they're going to pull or loosen these fibers, which are called zonular fibers or these suspensory ligaments. And as these are pulled tight and as they are kind of let loose, it changes the shape of this lens. So sometimes the lens can be kind of round like this. Sometimes the lens can be very straight, all right? And so depending on the shape of the lens, we are better suited to see things up close. So in the event whenever the lens is round, or we are better suited to see things at a far distance in the case of when the lens is more flat and more straight. Okay, so as these ciliary muscles contract, they pull or loosen on those suspensory ligaments. That changes the shape of the lens, and that allows us to see things up close or see things far away. So you can see here in this image here, ciliary muscle is relaxed. These zonular fibers or those suspensory ligaments are tight or taut, and that causes the lens to flatten or to become more straight, and this helps us see things at a distance. Likewise, if we see when the muscle is contracted, we have our zonule fibers or those suspensory ligaments be very loose, and when they're loose, the lens is more rounded, and we can see things up close. Okay. All right. So that process here, before we move on, this process of changing the shape of the lens to see things up close or see things at a distance, this is known as the accommodation reflex. Okay, and so you can test this pretty easily yourself. You can hold something right in front of your face, and you can focus on that. So hold your finger, your phone, or a pencil right in front of your face, and you can focus on it. Everything in the background becomes blurry, but you can see your finger, your pen, or your phone, whatever you're looking at, very clearly. You could then look past that thing in front of your face. So look past your finger into the distance, and now the distance becomes clear, but your finger becomes blurry. Okay, so this has to do with us changing the shape of our lens. So light is that is coming into our eye is focused a little bit differently, so we can see things more or less clearly, depending on if we're looking at things up close or if we're looking at things at a distance. All right, if we continue to look at our middle layer, our vascular layer, we have something known as the iris, right? So we've all heard of our iris. This is the colored part of the eye, right? So we have different genes that control the color of our iris, but the main job of our iris is to control the pupil diameter. Okay, so we know we have our iris out here all in blue. Here we have our round pupil, right? So the pupil is just an opening. Light passes through the pupil, and depending on the state of the iris, the pupil will either be very large or it will be very small. Okay, so there are muscles in the iris that are known as sphincter muscles. So there's ones that sphincter just meaning they surround this opening. There's ones known as dilator pupillae, and there are ones known as sphincter pupillae, okay, or constrictor pupillae. These are muscles that are either going to dilate, so they're going to make dilate the pupil, they're going to make it larger, or they're going to constrict the pupil. They're going to contract and close and decrease the size of the pupil. And these are going to contract, and again, these are smooth muscles. These are going to contract during different environments where we may have more or less light present, right? So whenever we're in a dark environment, and we're trying to gather as much light as possible, the dilator pupillae muscle is going to contract. The iris is going to get a little bit larger. It's going to kind of constrict out. The pupil is going to enlarge, and we are able to bring in more light. Likewise, when we are in a very bright environment, a very light-rich environment, our pupils are going to be smaller. So that sphincter pupillae or constrictor pupillae is going to contract, decrease the size of the pupil, and we bring in less light. Okay. So our iris is also a boundary, if you will, between our anterior cavity of our eye. So we'll see in a little bit we have what's known as an anterior cavity and a posterior cavity. So if you look at this image down here, we have all of this region here. This is all what is known as the anterior cavity. All of this region back here, this is known as the posterior cavity. Okay. In our anterior cavity, we have sub-regions. Okay, so we have chambers. So we have an anterior cavity, and then we have an anterior chamber and a posterior chamber. If you see, if I get rid of this drawing, if you see these dark structures here that I'm drawing in red, these are the irises on either side. Everything in front of it, this is the anterior chamber. Everything behind it, this is the posterior chamber. So this iris is the dividing segment between anterior and posterior chamber. And in this region, we have something known as the aqueous humor. Okay, and the aqueous humor is secreted and produced by those ciliary processes that we mentioned just previously. Okay, so it will circulate between this posterior chamber and the anterior chamber, and then it will ultimately be reabsorbed. Okay, whenever you have this aqueous humor circulate and it is not reabsorbed or it builds up, this is known as glaucoma. Right? So we've heard of glaucoma before. All right, so if we continue looking, we have our iris again. So remember, this is controlling the size of our pupil. It is containing smooth muscle, right? So this is involuntarily controlled muscle. These chambers that we just discussed here, and then again, the pupil is opening to allow light to enter the eye. So light is coming from the environment, passes through the cornea. Depending on the size of the iris, it goes through the pupil here, hits the lens, and then it is refracted to some part of the eye. All right, so here's a better view of those chambers, of those cavities. So in our anterior chamber, our anterior cavity, we have this region here, and again, this is containing all what's known as aqueous humor. Okay, this kind of clear fluid. It's similar to what cerebrospinal fluid is. The posterior cavity is all of this region back here, right? And so this contains something known as the vitreous humor, or the vitreous body, and it's this large kind of gelatinous structure that actually helps to keep the retina intact, keep it on the surface of the eye, and it also helps to maintain the shape of our eye as well. Okay. All right, so here are these muscles that we can see associated with our iris. So we said that there are ones that dilate the pupil and there are ones that constrict it. So here we see our dilator pupillary muscle, and here we see our constrictor or sphincter pupillary muscle. So whenever these contract, the radius or the diameter of our pupil is going to either get larger, it's going to get smaller. So if you look at this image down here, you can see that this individual's eyes are the pupil is undergoing a change in the presence of light, right? So as the light is shined into this person's eye, the pupil constricts, it gets smaller, so you would have this pupillary constrictor, or pupillary sphincter muscle would contract, and you would get this very small pupil. Or if you had a dark environment, it would relax, and you'd get this very, very large pupil that you see here on the left. Okay, these are autonomic responses, and so whenever our eye gets larger, or whenever the pupil gets larger, it dilates, that is a sympathetic response. Whenever it gets smaller and it constricts, that is a parasympathetic response. Okay. All right. So here you can see these. So this here was talking about our pupillary constrictor muscle. Here we're talking about our pupillary dilator. So you can see the pupil getting very, very large. All right. Now, there is a clinical sign, something you can look for when patients are taking opioids, and it's something known as pinpoint pupils. Okay, so this has to do with not necessarily the light coming in, but it has to do with the medications that they're taking. And pinpoint pupils can be a sign of potential toxic levels of opioids that somebody has taken, whether in a recreational setting, they're taking them legally, illicitly outside the hospital, or if they're receiving opioids in the hospital. You can still look at it, and so this is why you check patients' pupils relatively often if they're on an opioid medication. Okay. All right, so if we move to the inner layer of our eye, so we've gone from our fibrous outer layer, we went to our inner layer, or our middle layer, the choroid, right, this middle vascular tunic, and now we've gotten to our inner layer, which is known as the retina. Okay, so the retina consists of two main sublayers. One is called the pigmented layer, meaning it contains pigmented cells, and the other one known as the neural layer. And this neural layer, this is where we're actually transmitting the light signal from light to an action potential, and then sending it along the optic nerve to the brain. Okay, so if we look here, we can see, if I highlight here in red, this is our pigmented layer. Okay, and this is the back of the retina, and then all of this here, all of this section, this is all the neural layer. Okay, so the majority of our retina is this neural layer, and a very small amount is the pigmented layer. The pigmented layer contains pigmented cells that can absorb stray light and prevent it from scattering, like we mentioned earlier, to help prevent a distortion of the image that we're seeing. Okay? We'll see in a little bit when we talk about photoreceptor pigments that part of our photoreceptor pigment can be derived from vitamin A. And so you may have heard the term, or you may have heard the idea before, that carrots are good for your eyesight. This is because carrots contain carotene, which can be converted to vitamin A, and then we can use this vitamin A to make our pigment, our photoreceptor pigments that we store in our rods and cones. Okay, so the neural layer back here, the most posterior layer contains our photoreceptors. So these are our rods and our cones that we've mentioned before. One unique thing about our retina is actually the direction that light passes through it initially. So here's our pigmented layer, right? Here's our neural layer. Light is coming from here. So say your pupil was up here, right? So light passes through here, and it actually goes through this neural part of the retina, through our photoreceptors, and strikes this pigmented epithelium, and then the signal turns around and goes this way, and then goes along the retina, the neural part of the retina here. So it's almost kind of backwards as to what you may think in terms of the direction of light and the direction of the action potential. Okay, and we'll talk about these sublayers of the retina here in just a little bit. Okay. All right. So if we look at our light and we look at our eye, we know that light is refracted. So in other words, light is changing. It is changing speed as it passes from one medium to another. So what this term medium means, medium is the, you can essentially think of as the environment that the light is passing through. So if you think of like a spoon in water, like this example over to the right, or if you put a straw in water, it looks as if the spoon has been broken in half, right? So here's one part, here's another part, right? It looks like it's broken. We know it's not broken. It has to do with the light traveling through air. So here the spoon is in air. Here the spoon is in water. It's passing through different media, and so it is perceived by our brain as being different. It's perceived as being apart. So this is this idea of refraction, okay? So here we see that light, when it is refracting, it is traveling in one direction. It hits glass, or our lens in this case, and it's going to, or our cornea, it can then be refracted, hit our lens, and be refracted another way. So here we see light is coming in, it's going to hit our lens, and it is refracted, and it is focused on a central point. Now this central point we'll discuss, this is important, this is known as the fovea centralis, and this is where we have our best color vision, this is where we have our best, or a large majority of our cones and our photoreceptors that are sensitive to color. Okay, all right. So as we're coming in, again, we said that the lens is going to be responsible for focusing the light on a specific region of the retina. Okay, so here's this fovea again back here. So depending on the shape of the lens, right, it's going to be able to focus things good for near vision. It's going to be good for focusing things for far vision as well. And changing lens shape is responsible for this. And we can also know that we can change really kind of the shape of or the direction of light before it reaches the lens by wearing corrective lenses, right? So if we wear corrective lenses that are convex versus concave, we can have variations in the direction of the light and the way it is refracted. And so this is used for things like somebody who has myopia or hypertropia, right? So people who have, they're nearsighted or they're farsighted, right? So we'll talk about these in a little bit. But the thing to know now is that our lens is going to be focusing the light. So the big term here is focus. It's focusing the light coming from the environment. It passes through the cornea, through the pupil. It is then refracted and focused on our fovea, and then we have a signal that goes through the optic disc, to the optic nerve, and ultimately to the brain. Okay. All right. So here we can see those suspensory ligaments that can alter the shape of the lens. So if we're altering the shape of the lens, we can be better suited for near vision versus far vision. So the relationship between the ciliary muscle and the tension of the fibers is kind of counterintuitive almost. So whenever our muscle is contracted, whenever this muscle contracts, the fibers are actually loose. So when this ciliary muscle contracts, these zonular fibers or these suspensory ligaments become loose, and the lens is allowed to bulge, or it is allowed to become more curved. This is good for near vision. So I'm just going to write an N for near vision. Okay. Whenever the ciliary muscle relaxes, so it is kind of pulled backwards, it actually pulls on these fibers, so they become taut, they become very tight. That causes this lens to be more straight. Okay. When this is pulled straight, or it's more flat, this is good for distant vision, right? Or things we are seeing from afar. All right. And so again, it has to do with these ciliary muscles being contracted. And again, it's kind of counterintuitive. When the muscle is contracted, the ligaments are loose, and so the lens is bulged. Whenever the muscle is relaxed, the fibers are tight, and the lens is pulled flat and thin, and that's good for distant vision. All right. Okay, so this accommodation reflex, this is what we're talking about here. So if we're viewing things 20 feet or more, the muscle relaxes, ligaments become tense, lens is flattened. If we're viewing things closer than 20 feet, the muscle contracts, ligaments are loose, or the muscle relaxes, ligaments are less tense, and our lens becomes more round. Okay, so this is known as the accommodation reflex. Okay, so think about what happens as this occurs. So whenever our muscle contracts, it becomes very tense. Those ligaments are going to be less tense. They're going to be more loose. The lens is more spherical, so we can see things close. And then again, when it relaxes, the ligaments become tight. The lens is more flat, and we can see things at a distance. All right. Okay, so we can have various clinical impairments of our vision depending on the shape of the lens, right? So the term emmetropia, this is normal vision. Okay, so this is normal or whole vision. All right. So what you have is you have light coming in through the cornea, passes through the pupil, hits the lens, it is refracted, and it is focused nicely on our retina, and we're able to see a nice clear vision. Okay, so emmetropia is kind of our baseline starting point. This is normal. All right. So what we have next is we have hyperopia. So the term hyperopia means farsightedness, right? So the term hyper means kind of above and beyond. So this is our farsightedness, right? So what this means is that you can see things far at a distance, but you have trouble seeing things up close. So what this means is that our eye is actually too short. So where the actual fibers would, or the actual light rays would converge, would be at a point beyond the actual focal point. So the convergent point is distant, a distant point that allows us to be able to see things at a distance, but seeing things up close is actually made more difficult. So the way you treat this is you can apply, prescription eyewear, prescription glasses that have a convex lens, meaning they're curved outward. So this shape here is convex. Concave is when you have something like this, right? So this is a concave structure here. This is convex. What this does is it begins to focus the image actually on the actual retina, as opposed to where it would be focused beyond the retina. So this is why we see things very well at a distance if we have hyperopia, if we have farsightedness. Whenever we wear these lenses, so imagine the glasses here, the glasses are sticking out in front of the eye. The light's going to hit the lens of the glasses before it hits the lens of the eye, and so it is focused on a point closer to the actual retina, and this helps correct this distant vision. So if you know hyperopia, then myopia is exactly the same, except it's completely opposite, right? So now we have trouble seeing things far away. Instead of our eye being too short, the eyeball is actually a little bit too long, and the image is actually focused, as whenever we have myopia uncorrected, the image is focused too close, right? So here we would have the image being focused right here, when in reality we would want it to be focused right here, right? So this is uncorrected. Whenever we can apply a lens, now we're applying a concave lens, right? So we mentioned convex with hyperopia. This is a concave lens. As the light is coming through, it hits that, it is focused out, and then in again, and it hits nicely on our retina, and this helps to correct this vision. Okay? So again, this is where the glasses would be, this is where our corrective lens would be, and again, this is concave, right? So this is concave, whereas here, if we were talking about hyperopia, we had one that was convex. Okay. All right. So we are going to stop here, and we will pick up by talking about different layers of our retina. We'll talk about the actual neural and pigmented layer, and we'll look specifically at our photoreceptors, our rods and our cones, and how these serve to actually transmit an action potential toward the brain. But before we do that, keep in mind again, as we move on and before we get to the next part, light is going to pass through. So here's our optic nerve, right? So this is where the signal is actually going to be going to the brain. Light actually has to go through that, all the way through all these layers of cells, hit these pigmented cells, and then the signal turns back, goes this way, and then continues to the brain like this. So it's kind of opposite of what you may think. So keep this in mind, and we'll discuss this in detail whenever we get to our next part. All right.